Hello again, everyone. How are you? I'm very happy that we can continue our session in the podcast on drug delivery systems. So in this second session, we will talk about the delivery systems for proteins and nucleic acids. So why this topic is special? It is because mainly protein, because we use a lot of protein. So we know a lot of drugs called protein pharmaceutics. Protein pharmaceutics is basically proteins that we use to treat a kind of pathological condition or disease. Okay, so and then also come with the protein is the nucleic acid, because then they kind of have a same mechanism of action. Later we will discuss about it. Okay, so again, I'm Aditmika from the Laboratory of Physical Pharmacy and Biopharmaceutics, Department of Pharmaceutics, Faculty of Pharmacy, Universitas Gadjah Mada. And again, with me now,
I'm Ronnie.
Pak Ronnie is from the Laboratory of Pharmaceutical Engineering, Pharmaceutical Technology, the same department as mine, and the Faculty of Pharmacy, Universitas Gadjah Mada. And he has been doing a lot of research also in creating the systems or the delivery for protein in particular. But then later we can also discuss is to connect it with the nucleic acid, because again they kind of have same mechanism of action. So okay, I think it's quite interesting to discuss about the delivery of protein, because protein has a lot of aspects to think about it, but later we will discuss it one by one. So the first aspect is that protein as a protein pharmaceutics, as a drug, is like passwords. It is very potent, because it can recognize a very specific mechanism of action. If it's receptor, then it's very specific receptor. But the problem is that this protein is also a password for the same receptor in different cells. So this is based on your experience. If we want, for example, to deliver this protein, and we want to make sure that this protein is then transferred or delivered in the right targeted place, then what should we do?
Yeah, protein is for me, it's very special, because when we talk about protein, it's special because they have three-dimension form.
Yes. Okay.
Not just because when we talk about the drug or chemical drug itself, as long as the chemical, it's not
formula.
For the formula is not break or it's not breakdown, they still can have effect.
Okay. Yes.
But protein is quite different. For example, the amino acid of the protein is still the same, for example, but then they have a 3D structure is different or changing. The effect is completely gone. Or it's not different, but it's gone. For example, when we talk about
at least for the affinity, for example.
Because so they have like disulfide bond, and because they have something like that. So it's quite difficult. Protein have like you mentioned before, have very future advantages, but of course good and bad always hand by hand, always like that. You have a good aspect, also have a bad aspect. We call it barrier, for example, because it's easy to they have a problem with stability. For example, when you are working with insulin, for example, you have to store your insulin in the -4 or in the fridge, for example, to maintain the stability, because you cannot just put it in the room temperature, because like I said before, it's not break, but the 3 dimension is changing, so the effect is gone. So the big problem is I told you before is stability. Yeah, when we talk stability is one of the application that might help with nanoparticle, because maybe from the first session we talk about how to encapsulate drug.
Yes.
Protein also as a drug.
Yes.
So we can increase the stability of the protein with encapsulated that itself. I mean, with nano encapsulated of the protein, because and then they can still form 3D dimension that's still working when you apply, but also they cannot break down.
Yeah.
I mean, because protein is, for example, if you just inject it, protein maybe also can form another 3D dimension and then it's gone.
Yeah.
So, but the good things about protein is easy to handle. I mean, easy to handle means they don't have problem with solubility. So you can easy to formulate the protein
yes.
in different form. I mean, because we just play around with water, because its protein is EPCS 1 or I think,
something
and
like
then
yes,
one and then easy but the big problem is with instability of the drug.
Okay, so yes, so in the formulation perspective, it's pretty easy to handle because then the solubility is pretty good.
Yeah.
But then also with the stability, it has three-dimensional structure and things like that. And then we have to in the formulation process itself, that we have to keep this structure into this natural condition, so we can make sure that protein will still work. Okay, but then again, okay, maybe we'll go a little bit to the nucleic acid. What's the difference here?
Yeah, nucleic acid is of course is different form of the protein, because nucleic acid protein is consists of amino acid.
Yes.
But when we talk about nucleic acid, is consists of base.
Okay.
So they have and they I mean, when we working with let's say one of the example of nucleic acid is with DNA. Yeah, against DNA, first they don't have any problem with solubility because it's water-based. It is the RNA, DNA, or plasmid or something like that. It's the same, but against they have problem with with stability.
They are even more vulnerable, I guess.
Yes, because in biology we work we study what DNA. DNA is create. I mean the basic of the DNA is inside the cell.
Yeah.
So whenever they inside the cell, they still in good condition or in stabilized form.
Yes.
But whenever they outside the cell, that's problem. Because this is
we have to create the condition
how the naturals work.
Similar to the inside the cell.
Yeah, right? Because if the DNA is still stabilized in outside the cell, and then they can go everywhere, and then it's dangerous because they can enter everything and then can make mutation or something like that. So whenever they outside the cell, they will destroy.
That's
because DNA, when they enter to the liquid of the body, they will destroy, because the liquid is our body is full of nucleus.
Yeah.
This is
so we have to work
it's
in
biology,
the strict buffers.
It's how
temperature
natural is working.
Yeah, that's right. Okay, so basically the two are the same, and then they are also working in a biological system with the cell as targets, I guess, because for DNA RNA it's not possible outside the cells,
Yeah,
so they are
yeah.
working inside the cells.
Yeah.
Also with the protein, maybe their
Yes,
target
quite
is
the same,
also the receptor on the membrane. That's also possible, but
Yeah.
still it is on the cells.
Yeah, the cell, yeah.
So yeah, okay. So now our concern is just to deliver these two type of molecules, polymeric molecules
Yeah.
with all this structure, and then what approach we can do with this? I mean, we have to work very strict, right, with this. So it's even more difficult than before. It's not only about the polarity, for example.
Yeah, yeah.
But do you have any experience on working with this? For example, maybe vaccines, because it's quite interesting also, right? Vaccine can be protein, can be cellular matters, can be RNA itself. It's also possible. And I think it's also part of these topics. Yeah. So, do you have any experience about this? If we, in general, maybe.
Yeah.
Not yet talking about the administration.
First of my project when I did my PhD program is I working a lot with plasmid DNA.
Okay. Okay. And
because my goals is how to deliver the recombinant vaccine, for example, to our body. So at that time we use pcDNA or plasmid copy DNA that can express a gene inside the human cell. So we make some construct.
So it's just the same as virus then?
Yes, we make construct of the plasmid that consists of some gene that can enhance or can trigger the immune system, for example, for immunization or to vaccination. Yes, it's the same. The problem is when we try to inject just naked plasmid, it's easy to degrade because that plasmid have to work inside the cell.
Yes.
The question is how we deliver the plasmid inside the cell, because before you enter the cell, you have to face the liquid of the body that consists of nuclear cells that can destroy or degrade the plasmid itself.
Yes.
So, again, the answer that time also till now is nano. Yeah, I try to have different type or different application to working with to protect the DNA, but it's not working as well as nanoparticle. So at the time.
Okay, so you did try another systems.
I try just maybe to just add in some protection or something, but still it's not working. Okay. Still, the plasmid is degraded easily when they start contact with our body, because it's like it's natural. You cannot against the natural, because you can against the nature because how the way it is, you can.
Yeah, but then to think about it, it's quite amazing to think that virus can really create pandemics, right? Because they need to be transferred.
It's virus is smart, So, it's small, and the good thing is virus is like smart because till now maybe a lot of discussion. This is life or non-form life or non-life form. Still, but it's working.
Okay.
So virus also, they have they have capsid and that envelope to protect their the DNA, their DNA or RNA.
Yes.
For example, now we fighting a lot with COVID-19. Consists of this COVID-19 is RNA virus, so they consist of RNA, but they have a capsid on an envelope to protect their DNA or the material, genetic material. So, in general, you have to protect the genetic information, DNA, RNA, or saRNA, and then whenever they enter the cell, they can still they will working. Yeah, like virus.
Yeah. So
Virus
did
is
you
kind
then
of-
mimic how virus works?
Yes, yeah.
At the
Virus
time?
is kind of delivery system.
Okay.
Yeah,
Wow,
because
that's
they
interesting.
deliver
Yeah.
their material genetic inside the cell. Because virus is like I told you, is non-living thing, you know.
Yes,
But
yeah.
whenever they enter the cell,
Then they are
they will
yes.
replicate, you know.
Yeah.
So like we know about bacteriophage, for example, they will replicate inside.
right.
So how it works is the same like virus. But of course, virus is God creation, you know. I mean, it is very perfect
Yeah.
and maybe it's not easy to mimicking,
mimic,
but
yeah.
we can learn about how they work.
Okay.
Yeah, they protect their genetic material. The same like plasmid that we try to make a vaccine, like I told before. We also encapsulate the material genetic with in this case we use chitosan because.
So at the time you use
Chitosan
okay,
because we're dealing a lot with because how to make nanoparticle is especially with DNA is with ion chelation. As we know, DNA is negative charge because they have big bond phosphate. They have 4 O. So they have a highly negative charge. So whenever you want to make another particle with DNA, you need counter ion of the negative charge. So you use positive charge polymer. In this case, I use chitosan that I mentioned before. Chitosan have positive charge. So just combine it.
Yeah.
Yes, positive, negative. We just put it together. They will like we make encapsulated by their cell. Whenever they encapsulated inside the nanoparticle, they protect it from enzyme.
Okay.
Because the enzyme somehow cannot find the target because the target is hiding inside the nanoparticle.
In the matrix, yes.
The good thing also, as I mentioned before in session 1, that cell is negative charge.
Okay.
So
same. Then again,
the polymer is the chitosan the nanoparticle is positive charge, so they can attach to the cell and then endocytosis.
Yeah.
So whenever they enter the cell, and then whenever the fire endocytosis, the particle
is
and then
broken from the fat wall.
Yeah, because they have endolysosome,
yes, that's
so
right.
they can break and then unload the plasmid into the cell.
Wow. Actually, it's quite amazing to think about that. The chitosan,
this chitosan
actually
is for
mimic
me
sort of mimic the function of the plasmid. Okay, but then since the polymer itself they have charge, right? Can we confirm that this polymer will not alter the properties of the DNA, RNA, or protein that is encapsulated?
Yeah, of course. This is our concern, of course, because if you have a more charge, this is also can destroy. You know, for example, you have electricity. You know, it's also dangerous. You know, but of course, and then we manage that how because the charge itself we need it, but not so much. So it's like compromise. So the good things about chitosan is it came. I told you before, it came from nature, so it cannot be harmed.
Yeah.
So first it's safe for sure because it came from the nature. We call biopolymer, but of course to increase the capability we need effort, for example. Not just make nanoparticle. Also we increase the stability with adding 3-polyphosphate, for example, to make cross.
Okay, so then S particle itself will become stable.
Mm.
Okay, nice. So I think now we we'll go to answer the question,
Yep.
right? Okay. So there's a question from our students. So yeah, there's one, this question, I think it's interesting. It's about the edible vaccine.
Edible,
Yes,
orally
so
vaccine.
it's, yeah.
Okay, yeah.
If you want to deliver this protein, because you know you mentioned a lot about this stability, okay? And then, well, we're still talking about something that is injected intravenously.
Yeah.
It will become more complicated, of course, if we need to deliver it orally, right? And then, I think you already have done research, right, about this edible vaccine or something?
I mean,
in the past?
yeah, of course, oral delivery system is inconvenience, you know.
Okay.
I mean, we are pharmacists, isn't it? Yeah. So our goal is always to make the patient comfortable.
Yes.
So everything that we want to
Sure.
make, just to make our patient or our customer is comfortable. I mean, user friendly, for example.
Because it will also improve their compliance.
Everything,
right?
yeah. Improve compliance, and then easy to eat, for example, easy to drink, and then they increase the compliance, and then the effect of the dose is increase that blah blah blah.
So, for example,
oral.
Is it
Yes.
possible to deliver proteins or nucleic acid orally?
is challenging, but when I mean 4 or 5 years ago, I have a project. I apply a project with not vaccine maybe, but insulin for oral.
Okay.
I
Yeah.
mean,
Okay. Insulin is
quite
also
the same.
protein.
I mean,
Yes.
yeah, it is the problem is quite the same. At that time, a lot of people say that, is it impossible, Ronnie? I mean, like,
because insulin is something that you need to
it's protein,
take
you know.
in daily basis, right? Because
And then insulin is easy to degrade it when you use for oral.
yeah, of course. Yeah, I mean, you don't want to inject
Yeah, of course.
yourself
Yeah, inject-
all the time, right?
Yeah.
And then you want to do it orally.
Yes.
But then, is it possible to have this insulin?
Some people say it's the question is also some people ask me, is it impossible? It's like science fiction, you know. I mean, like, of course, when we just swallow or doing orally for protein, in this case, insulin is easily degraded.
And then,
Simply.
what would we do in that case? Is it impossible or still possible?
And then I get the project for 3 years, doing some research, and again, the answer is nanoparticle.
Okay.
Because the same story about nanoparticle with insulin, because the issue is how to increase the stability inside the GI tract. First, they have to protect from the gastric juice, because the pH is also the problem, because protein is easy to
acidic,
degrade it, and then when they escape from the gut, and then they will face problem in the intestine, small intestine, because there are a lot of enzyme, protease, and so on inside the intestine. Then
And
can
we can
easy
also find cytochrome P450 there,
So-
right? So
the answer simply is we have to protect protein till the target.
Yeah.
So, and the answer is against nanoparticle. And because, a lot of people say this is impossible. Let's try. Of course, science is about try and error. It's not.
But based on your experience.
And then we do research for 3 years until in info study. At least we can prove our concept.
In vivo.
In vivo.
In vivo. Okay.
In vivo.
Nice.
So we create our nanoparticle with insulin nanoparticle with some
And what's
special.
the materials that you use for
I use
nanoparticles?
because I like to have biopolymer a lot.
So you use
I
polymers.
combine chitosan and pectine
Pectine,
and alginate, to cross to make fine nanoparticle. Finally, I can make fine nanoparticle, and then because we're working with protein, and then I have to freeze dry it, because protein is cannot easy to spray dry, for example, because it was broke because of the
high temperature.
temperature.
And then,
I use freeze dry, and then we make a powder of it, and then put it in capsule, and small capsule, and then we're doing in vivo study with mice, or not mice, rat.
Rat, okay.
That we already destroy the beta pancreas.
Okay.
I mean,
so then basically they are diabetics.
Diabetic,
Yeah,
to make a diabetic model rat.
Okay.
For example. So we make it, and then we put it in, and the condition of the rats, because they don't have insulin inside the
Okay, yes.
and then we give the rat orally with our insulin nanoparticle, and then we of course the control is without nanoparticle or just insulin.
Yeah.
And then we combine. The result is quite interesting. When we add the rat, because before after we give the rat with nanoparticle insulin, we monitor the concentration of the insulin in the blood, of the serum of the rat, also the glucose level. So in some point we check every time. So because when we add the rat with naked, we call it naked, insulin, we control the insulin. We check the insulin by time. We see the insulin increase in 10 minutes because the concentration is still okay, but after 10 minutes it decrease. It means they start to degrade it and flat.
Yeah.
But when
okay. Yeah. So you measure the insulin or the glucose inside the blood?
Both of them.
Both. Okay.
Yeah.
Okay.
So, but maybe I will explain the insulin first, and then with nanoparticle is increase and still increase.
Okay.
So in this concept, we prove. I told you that I prove our concept that insulin can enter to the blood.
Yeah.
Is it amazing? That it was, for me, it was wow, you know.
Yeah.
Because whenever insulin getting inside the blood, it means they can escape from GI tract.
Yes.
With our, with encapsulated with our theory, they can. Is it wow? You know? So, and then to make our results more pronounced, we also check the glucose, because the question is the insulin still working, functional to decrease the glucose of the rat? And then we also maintains or check the level of the glucose and also decrease. So there is like cross section in the graph. Whenever the glucose is decreased, the insulin increase. There is a cross section. Is it like, of course, is not answer all the question, but at least that our concept is
very pronounced.
I mean,
proof of concept.
Our proof of concept is
that you can show
working.
that we can deliver proteins per oral, and then
of course, and then the question is
arrive at the blood.
Yes, this is the I mean also the same problem when we also try to make deliver for the DNA, for example.
Yeah. Okay.
So this is possible, but still we working on it because it's not. It's still working. Yeah.
And again, about the application of this delivery system, maybe nanoparticles is part of it for the protein and nucleic acid. For example, for vaccines, some vaccines, as for example this COVID vaccines, we need twice of the injections at 2 point of times. But is it possible that we create this kind of system that, for example, we can keep this vaccine to trigger the antibody for a longer period of time, for example?
I don't know.
So then we have to protect it. Can we, in theory maybe, protect this nucleic acid or proteins to stay inside the blood without any, biological metabolism, for example, from the macrophage or things like that.
Yeah, basically when we talk about vaccine or doing vaccination, you want to trigger our immune system to wake them up, to prepare for the battle, because the enemy came from outside our body, because everything inside this body is still ready to combat, But yeah, immune system is not that easy, I mean, like it's very complex, and it's not about just the drug and how you live, how you eat, and how your condition, is it good or not?
I mean, of course.
Yeah, I mean, you also have mentioned before.
And the virus itself is something that is still well studied, it's not yet known everything about the vaccine, virus.
Yeah.
And there are also have this kind of mutations.
Especially when you have to fight with RNA virus. Is never-ending story. I mean, like now with COVID, they mutated every time. So the question is, is our immune system is ready or not? Because they have to change every time. When you prepare your immune system with special targeted, for example, this is working for sure. But when the target is changing, they don't know. They don't recognize the target. So I mean, it's complex. I mean, depend on what are you fighting for.
Yeah. Okay.
COVID now we have a lot of evidence that like India, they say that the virus not just double mutated but triple mutated.
Yes.
I mean.
Okay. So in brief, delivering the vaccines to work well to make it to prolong its effect, it's something that is also
I mean, it's complex. It's not just single shot, but maybe multiple shot or multiple dimension.
Okay.
Yeah. For example, when we talk about virus, flu virus.
Okay.
Till now.
Yes.
We don't have
basically make ourselves strong enough for it.
Or maybe in Europe or in another country.
Yeah, I heard they have
every year they have to shoot
vaccine A, vaccine B, vaccine C.
Yeah, they're changing a lot.
Okay.
So it's complicated.
Yeah. Okay, so then let's make it in a more simple discussion. Now I think part of the interesting thing to use protein and nucleic acid in pharmaceutics is for example, if we want to treat cancer, chemotherapy always comes with a very severe side effect, right? Then we have to prepare that our hair
and also not suitable for the patient.
That's right. Okay, and then a lot of studies has proven that we can use nanoparticles to deliver this maybe protein or nucleic acid to correct or to kill the cancer. So, what is your experience about
cancer? Cancer is difficult. I mean, when we talk about cancer, is for me it's like something that we seem like we fight something that we don't know the reason. For example, if you have a cough, for example, or you have like inflammation in your throat, because of the bacteria or something, inflammation. Simply, if you go to the doctor, because the doctor knows about the cause of the cough, because of the bacteria or infection, they simply give you antibiotic, and then it's gone. Okay, but with cancer, what's the reason? What cause of the cancer?
Because if it's something that, I mean, oxidants, for example.
A lot of people say, with smoking,
sometimes
for
it's
example.
simply idiopathic.
I mean,
for one person it comes up, other person is
No.
Okay.
So it's the reason why cancer, when we talk about cancer, is lifestyle disease. So something wrong with your lifestyle, and then
Okay.
medication is not the answer. It's my opinion. But of course, we not give up about it. Of course, we still. So when we try to kill the cancer cell with our drug, for example, the key is how to find the right cell. For example,
Yes.
because the medication now with chemotherapy or with sinar gamma or something, gamma ray, is the problem is they kill all the cells.
Yes. Yeah.
So
is that possible to recognize
Of
or to distinguish between the
of
cancer
course,
cells
everything
with
still-
the
still-
nanoparticles approach, for example?
still possible because we can create nanoparticle with special moieties
Okay.
that maybe can target the cell
Okay.
with special antibody that can recognize the antigen inside on the cell, on the surface of the cell. They can, but yeah, maybe in my opinion, yeah, in my opinion, still difficult because till now we don't know exactly what's the difference with normal cell and cancer cell.
Okay. But I think, if I'm not mistaken,
Yeah.
then
This is my opinion.
but you have ever had a project, right, using the
Yes.
AdCam or something? If I'm
AdCam,
not-
yeah.
yeah. And then how is the
But-
result?
Yeah, for example, I have a project maybe with you. I think with
This one,
Prof. Sosimdari
Yeah,
and
Prof.
Pu Hilda. We try to create nanoparticle with special targeted therapy.
Okay. Yeah.
We use
MG protein, right? If not,
yeah the
Yeah.
the
but that's
drug
also
itself
protein, right?
is-
I mean,
is
yeah, that's also
protein.
an example.
The drug is good. They have potential. They have can kill the cell. For example, have a good capability to kill the cell. But against the problem is how to deliver to the right
Yeah.
cell. So in that project, I use cancer, special cancer. What called payudara?
Yeah, I think T470. Or,
Yeah,
something
yeah,
like that?
it's special. Yeah,
Yeah.
because they said
Or MCF7.
MCF7,
or something,
yeah,
yeah.
MCF7 for breast cancer.
Yes.
Yeah, I use that type of the cell culture. But the problem is, like I told you, there is no big difference between cancer
Okay.
cell.
So basically, it kills everything.
No, I mean, when we talk about cancer, if you read in journal, they say that this cancer cell overexpress that kind of
Something.
protein.
Okay, receptor. Yes,
But
protein.
the problem also, the normal cell also express the
the same.
Yeah. protein. So there is no big difference. Yeah, the difference is just this is a lot, this is less. But still they have.
Yeah.
So
Yeah.
the
And the
effect-
protein itself also works, if I'm not mistaken, the MJ protein
also normal cell.
with the ribosome inactivating protein or
Yes.
something like that.
Rib- yeah.
But basically all
the cell
cells
is killed.
they have ribosome, right?
Yes.
if
Yeah,
it's,
this
for the healthy
difficult,
cells,
difficult,
So
difficult.
then you try to create the
Yeah.
delivery that can select.
Yeah, of course. Till now, a lot of almost of scientists in the world
Yeah.
rising together
So if
to that goal.
So even we haven't
it's
talked about the
difficult.
stability, we still talk about the effectivity.
Activity,
It's
yeah.
already difficult.
Cancer
to-
is difficult
so,
for me. I mean,
yeah,
like
even in terms of
till
targeted
now, there is
delivery,
no
not possible.
therapy that suitable for the patient.
Yeah,
Yeah, we have a lot of
So,
story with our
we have to
friend.
endure the patient.
Yeah.
They have to endure the
Because cancer is like
side effect
like-
of yes.
you know
don't know reason
you
why.
don't know
Yes.
exactly why,
Yeah.
and then tomorrow
We only talk
yeah.
about probability.
Yes,
Okay.
So, yeah, of course, cancer is challenging, but I mean, it's difficult to try to fix it or to I mean I have experience working with it, so it's the reason we have to still fight that cancer, but until now we don't know the reason why the cancer happened, or why the cell changing normal to cancer. I mean.
Even if we can deliver, for example, nucleic acids to correct the DNA, we still don't know which DNA needs to be corrected and
I mean,
DNA of
now
which
I have a project with Faculty of Medicine in Gadjah Mada University, Universitas Gadjah Mada, also working with Prof.
Mustafa?
No.
How is it about? What is it about?
We're working with siRNA, small interfering
Prof.
RNA.
Sofia Mufarika.
Prof. Sofia, sorry, I forget it already.
Yes.
It's because siRNA also against they don't have good stability. They easy to degrade RNA. You know, you working with RNA is easy to degrade it. Even when we isolate and then in some hours they will gone easily. So siRNA is kind of RNA but small interfering that can how it work is blocking mRNA to make protein. So when we already have a lot of protein that can, we mentioned that protein can change the cell become cancer, or to
improve the probability of a cell to go into a mutation. So it can block the mutation.
Or to block the capability of cell to doubling.
Because the siRNA is basically interfere, right?
Yes, it blocks. So when they block or attach to the mRNA, they can make protein.
So in theory, you need to deliver this siRNA inside the cell. Inside the nucleus.
No, inside the cell.
Inside the cell.
In the cytosol.
In the cytosol is enough. And
because ribosome is.
So it blocks. Just in the cytosolic area.
Nucleus inside the nucleus is DNA. And DNA produce mRNA.
And then it will go out from the nucleus to the cytosol. And then So
before they come to the ribosome to make protein, they block.
Then you block with siRNA.
Cannot produce the protein.
So how is your strategy in that project?
The same like the problem is how to deliver to the cell.
And then
I'm working a lot.
Did it work?
It's work. It's good. And we have a lot of publication out of that. And now we apply for big project with KB Pharma.
Nice.
Because they invite us invite our group to make a product of it. So last time we have a lot of meeting because it's promising, you know. We deliver the siRNA to the cell with chitosan, because the same story, because chitosan is positive charge and RNA negative charge, we can easy to make a nanoparticle. And it's easy to scale up, and then of course we still working on, because in this project we do a lot of experience of the but still in my opinion we still have to find the right cell to deliver.
So
I mean this is the big problem. So to deliver is okay, but to find the right person, the right cell. Is
because if you deliver in the wrong cell, then it will do the same.
The same like chemotherapy did it.
Then you will
they will kill every cell because they will block. I mean this is This is the game, you know. I mean, yeah.
But I heard a lot that chitosan has a quite pretty general affinity to protein, for example. Then is it also possible for you to attach antibody, for example, in the surface of the?
Yes, yes. I think we also prove that concept also because we use ADAC reaction. I mean, we use ADAC. Maybe if you want,
yeah,
I mean, ADAC is like still very technical, you know.
Okay.
ADAC kind of like.
But can we do it in the normal circumstances in the lab?
Yes, yes, because it's just add-on.
Okay.
Just add-on. So we basically put it together between nanoparticle or chitosan, who consists of nanoparticle. Nanoparticle consists of chitosan, and then add the antibody and then put the ADAC. That's it.
Oh, ADAC is basically like a glue.
Yeah, like let's say it's just very technical. I don't remember.
Okay, so but yeah, okay. So it's like the link between the chitosan and the protein.
It's basically this one. So because chitosan have because the positive charge of chitosan is from NS group. Yeah. So we combine and that NS group with.
Oh, okay. So
with something from
so the
chitosan,
has
a moieties that can bridging the or combine with antibody that can attach to the
Okay, so that is also possible.
It is possible, but
have you tried with your project?
Yeah, I try.
But then, okay, so maybe you have difficulty on finding what kind of receptor is correct.
Yeah,
I mean, you can always find the anti-protein, right? The antibody.
Yes.
But then, you have to really sure about
the cell.
What? Yeah. Okay. And because then it's challenging.
Still. I mean, every scientist, I think, in this art. This world.
And then if we talk about mutation, maybe per patient it will
is different.
But for example, if we, just kind of a crazy idea, for example, can we just apply it? For example, if we design this application this is special delivery for patient A, and I also can create special delivery for patient B.
Yeah.
So just one design per patient. Is that then work?
Yep. Basically, it's working.
Okay.
Basically, but
so if we just forget about the cost, is it possible?
This is possible.
Okay.
Possible, but still, you know, technology is about how you apply.
Yes, of course.
I mean, we cannot just
so
say.
The challenging part in theory is that
of course, you cannot say
to create
like this.
Something that can work for everyone.
Okay. Because you know, will be impossible. For example, if you want to apply that kind of project or the kind of idea, and then the cost is
very high.
And then who will buy? You know, I mean, and even till now, you know, with that our technology, still we're fighting a lot with cancer. Because back again, like I told you before, we don't know how it costs.
I also heard a lot that the result is quite erratic. Like, you know, it works for this very, few people in this, maybe with the similar genetics, and then for other person in other country, for example, it is not working.
Because you don't know exactly how the cell changing from normal to cancer. We have to answer that question first. Before we
can find a targeted approach for this.
Because and then we found the causes of the cancer.
Okay. So now, basically, our main concern about delivering protein and nucleic acid is what we can do now is only just about the stability. So we can
of course improve the
stability. We can improve the penetration, for example. But we cannot be sure that this protein or nucleic acid is delivered.
Also, the big problem, I mean, cancer for me is still away, you know.
Okay.
But against now about vaccine.
Okay.
We have in this world now, when we talk about COVID-19, a lot of vaccine that we already produced from US, but I think one of the vaccine from US, as I remember, they have a problem with cold chains.
Yeah.
Because they have to store the vaccine in minus 20.
Yeah. And it is not,
and if we want to apply, for example, in our country, in Indonesia, for example, how it works?
Then the cost is very high. Yes.
Very high and nearly impossible for me.
Yes.
You know, when you have to deliver the vaccine inside, the vaccine to the Borneo, for example, is how you have to bring refrigerator, not just freezer, for example. That is, I mean, it is a big challenge for us to increase the stability of the protein or the vaccine by encapsulated.
Yes.
Because I also have a project with biopharma to increase because biopharma have a vaccine that also the problem have a problem with minus 20. So we try to
even for them, I mean, they have a lot of experience of producing this vaccine.
Because
till now they still have that kind of problem. I mean,
now it is big problem. Because the cold chain is big problem to vaccination.
Maybe the big portion of the cost then is for the delivery. I mean, the delivery, the transportation of the vaccine to the hospital.
I saw one movie from National Geographic about how to deliver vaccine from, I think, from India to Nepal.
Yeah.
So it's a long story. You know, you have to use airplane, car, and then horses, and then walking.
Yeah.
And because the villages just near the near the mountain, so and then they bring a lot of ice and then cost a lot of thing, a lot of money. So
maybe the vaccine itself is very cheap.
So cheap.
I mean,
how
but
to deliver.
Only the transportation is quite.
They're fighting with temperature and with the time, so it is difficult.
Maybe the better idea is just to bring the patient.
But I mean, also it's not difficult. It's not easy. I mean,
it's like a mess.
A mess, I mean, a lot of we still have a lot of problem with the protein and the vaccination or vaccine in general. So
and in Indonesia
also have need it.
1,000 islands.
1,000 islands. So if we can find solve the problem, I think is good for us because we can solve the vaccination.
Okay.
Because
but for example, if you have an opportunity to just start, then what will you propose? What kind of encapsulation?
Yeah, well, we try. I try a lot with nanokitosan, and that time because, for example, the vaccine from minus 20, we can make that vaccine stabilize in 4 and 8.
4, okay. That's good enough.
I mean, still in one shot. I mean, I have to work a lot. But
I mean, if you can make it in 3 days, just in an ice pack, for example, then it's already
it's good.
A lot of improvement.
Because we just can can just use ice box to carry on.
Yes.
I mean.
So actually, there are a lot of efforts already started. On that direction.
Yes,
Okay. So
because the issue is always stability to deliver the drug, also stability to deliver to our body.
So there are two different
still delivery system. I mean.
Because if we talk about production, industrial.
And not so much people, not so much scientists in our country working in delivery system. I mean, of course we.
That's correct.
I mean,
they usually focus on the pharmacology or the compounds.
Compound.
But not the delivery
system.
Then,
I mean, because for me, when we're working with delivery system, it's easy to start because we have a lot of problem with delivery system. When you compare to find another compound, for example, I mean, a long way to go, you know. I mean, like you told me that when one single entity drug will need a lot of time, a lot of money to have license, for example.
Yes.
So
And then, yeah,
and
of
then
course.
yeah, like I told you before that paracetamol still exists till now.
Yes.
I mean, of
Maybe
course,
that's the easiest.
a lot of studies say that paracetamol is hepatotoxic, blah 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 blah. But now, still we have paracetamol and still use it.
Okay.
I mean, yeah, it's just
Yeah. So if we can summarize our discussion, that maybe for the last part of the discussion. Okay. So first, then maybe we can be sure that this nanotechnology to deliver protein or nucleic acid can at least improve their stability. Is that correct?
yes.
Okay. From that point of view, is it also possible for us to deliver protein per oral?
Yes, deliver.
That is also possible.
Possible.
You already proven
Yes,
with the
but still
experiments. Yes, of course. But yeah, there are. I mean,
On
a
progress.
clear path
on the way.
on there. Yes. But then, if we talk about targeting, it's something different.
Yeah,
Okay.
something different.
Okay. And then another thing that makes this stability important is that because of the transportation of this drug. You know, if we create vaccine or maybe later protein pharmaceutics, then we need to deliver in a very strict condition for the stability. But if we can create the encapsulation in a right way, that this distribution can be more effective, efficient, then we can cut the cost.
Yes.
Okay. Yeah, well, it's really a nice discussion with you.
A lot of discussion, and then
even for me, it's also still a lot of information that I got. Something new. So for the students, I think that's it for the second segment. And we'll continue with the third until fifth. We still have some interesting topics to discuss about the drug delivery system in the next podcast. And that's why, again, if you still have any question, I've already given you the link, so we can discuss in our next session, in our next podcast, so then it will become more beneficial for you, because then the application is then as what you wish to understand. Okay, again, thank you very much, Pak Ronnie, for the discussion, and thank you very much for the students for your attention, and see you again in the next session.
See you, everyone.
With different topics and also source person, of course. Bye.
Bye.